All right, so as we get through the latter part of chapter 12, there are a few key terms that I want to share with you. And one of them, it's a fun term. You'll see when it comes up in a little bit. Antagonistic pleiotropy. Antagonistic pleiotropy. And so this is a term we saw back in chapter 9. It basically is addressing that the impact of a certain mutation is condition dependent. And what I mean by that is the impact of a particular mutation may be dependent upon the sex of the individual, or the age of the individual, or the overall health of the individual. And so antagonistic pleiotropy is basically describing mutations that early in life have very little impact, or may even be beneficial to the phenotype, but later in life might become a big complication. And in a way, it's an attempt to explain several specific mutations, several specific alleles that correlate with certain complex genetic illnesses, and explaining why natural selection hasn't eliminated them. And it's because early in life, before you reach sexual maturity, or even at sexual maturity, it has either little impact on the phenotype or is actually beneficial for the phenotype, but then later in life makes you significantly more likely to develop a particular genetic illness. Okay? And I'll give you a specific example of that in a bit. Genome-wide association and interaction study. It's a mouthful. Maybe a couple mouthfuls. Couple mouths full. There we go. Grammatically correct. So we've talked about genome-wide association studies, and this is basically, bless you, where you're looking at the entire genome and looking for any place that correlates with some specific phenotype, with some specific character. Now, with these genome-wide association and interaction studies, we're looking at basically that same thing, any part of the genome that correlates with a particular phenotype under different environmental conditions. Okay? And so we do these when we have a suspicion that, one, there's some genetic aspect to the development of this phenotype, whatever it is. Let's say it's type 2 diabetes, that we have a suspicion that there's some genetic aspect of that, because it runs frequently in some families and appears very seldom in others. And you're like, man, both these families eat like Americans eat, which is not great. And so it's like, why is type 2 diabetes so much more common in this family than the other? Well, it's got to be because there's some genetic component, right? And so we go searching, we go hunting for the portions of the genome associated with the development of that genetic illness. And what we do is we design our study to basically have also take into account different environmental factors as well. Okay, so we can account for those. Were you going to ask, how do we do this? Are we going to do a controlled experiment, or?
No, I was going to ask, how do they know that the unifying thing between the families isn't some environmental factor?
Right. And so that's where we also have to take into account a number of environmental conditions as well. So a lot of times these genome-wide association and interaction studies are done as retrospective studies, where they'll recruit people with the genetic illness, they'll recruit people without the genetic illness, and then they'll basically look at everything in the lives of both groups. They'll look at their entire genome, okay? Looking for places in the genome, specific mutations that correlate with that genetic illness. And then they'll also take in an enormous amount of environmental data. I mean, they'll give these people ridiculously long and extensive surveys. And so you can find some of these, where you can see some of these surveys, hundreds of pages long, taking you several days to complete. And it's basically like, it's almost anything you can imagine. And then you have to run these really complex, robust analyses to find any of those independent variables that are going to correlate with the development of that genetic illness.
So can you repeat the first sentence of the definition?
Of the genome-wide association and interaction studies? Maybe. I'll have to remember what I said. And so what these are studies that look at what specific alleles correlate with the genetic illness while also looking at environmental influencers of the genetic illness. You bet. So it's basically taking anything as a potential independent variable. Specific allele, specific environmental experience. I don't even know, I should just say variable, specific environmental variable. I always want to use a different term, and I feel disappointed with experience, and I don't know. I don't even know what other term I would use. Nutrigenomics. This is a really fun term. Might be one of my favorite new terms. I don't know. And so basically, what it is it's an effort to look at how do specific foods impact people because of their genetic programming. Okay? How do specific foods impact people because of their genetic programming? And we'll talk more about this in a little bit.
The genetic programming of the food, or?
No, of the individual.
Okay.
Yeah. Yeah.
So it's like how different people.
Yes. Yeah. To where, I feel like I function best eating Taco Bell, but not everybody feels that way. And my wife doesn't really let me eat it very often because she wants me to live long enough for our kids to have kids. So sweet. So sweet. Oh, I don't know. What are you going to do? This one. This one is actually my favorite key term in here. Fauvism. Fauvism. And this term, it really has, I was going to say it has very little to do with the chapter. That's not true at all. It has very little relevance to anything except itself. And so what Fauvism is it's a severe allergic reaction to fava beans.
Whoa.
Yeah, fava beans. Also known as, there's another name for fava beans. I can't think of it right now. Fava beans are fun little legumes. So, you all know that cashews and peanuts are legumes as well, right? And so, biologically, but are we okay? Anyways, biologically they're beans, but they're so dense that they have a texture like nuts, right? And so fava beans are kind of somewhere in between, like what you typically think of as a legume, a bean, and something like a peanut and a cashew. And anyways, they're nice little beans. But this is one of the few things talked about in this chapter where this is a monogenic genetic illness, Fauvism. There's a mutation that when you have this mutation, you are always severely allergic to fava beans. And you're like, that's so cool. Because not a lot of food allergies actually work this way. Most food allergies are very complicated. They are complex, if you want to call them genetic illnesses, which you totally could, they are complex, multifactorial genetic illnesses. But this one, it's not. It's monogenic. And it's like if you have that mutation and you eat a fava bean, it's not great. It's not great. And on that note, if you all can think, if you all can take some time to think about my son, he's 5, and he's got some severe allergies to the pollen that's out right now, which is like one thing if all he's doing is like sneezing, coughing, whatever. It's like, dude, just relax, you'll be fine. But his entire body now is breaking out in hives, and his face started swelling today. It's pretty gnarly. So, and it's one thing when you're an adult and dealing with that, and you're just like, man, this is really annoying. But when you're 5 and you're dealing with it, man, it's awful. So, anyways. And he got sent home twice this week by the school nurse. This time it's like, you need to come pick him up and probably give him some Benadryl or take him to the emergency room because his face is swollen. So, but anyways, his name is Liam. You should pray for him. All he wants to do is watch me play video games. He's like, Dad, I don't feel well. Can you play video games and I can watch? I'm like, of course. And then my wife's like, Matt, stop playing video games. I'm like, he's sick. And he wants me to play video games. What am I supposed to do? All right. What do I play? Is that what you're asking? Mostly just his Nintendo Wii.
Yes.
Yeah. So he wants me to like play Mario Kart and just slay the other racers. He likes me to play the games that he can play, and he's 5, but because I'm better at them than he is, and so he likes to, I don't know, I think train himself by watching. So, anyways, sorry. Sorry. Wow. Oh, okay. So that's fauvism. That's really all we're going to talk about it. You know, it's done. Systems, biologic methods. This I put on there for a couple of reasons. One, the English is horrible in this key term, which I love it. It should be like biological systems. The other thing is it's basically what this key term is pointing out, and what they're trying to get you to understand with this is a lot of this work when we're studying gene-environmental interactions. And especially when we're studying gene-gene environmental interactions, it takes collaboration from all sorts of different kinds of biologists. Okay, it takes work by nutritionists, by physicians, by geneticists, by molecular biologists. Because the thing is, we know now, unless we're talking about like a monogenic character, it is super complicated to try to figure out whether or not just because you have a specific allele, are you going to get that phenotype? That's really difficult to understand, but it's really important to be able to predict. Okay? It's really important for the health of an individual, and it's really important just for healthcare costs in general. Okay? Which is basically a burden shouldered by everyone. And so, And so what this term is getting at is we're at a point now where we have the incredible ability to predict certain diseases and to prevent them, okay? And we have an ability to do this like probably no other time in human history. But we also have an incredible responsibility then, if we can do that, to actually do that. Okay, to be able to get really good at predicting not just if, but when you're going to start to experience the pathologies, and to try to push the pathology associated with these illnesses back. Okay, so that's what this term is getting at, is basically we're at a point where healthcare is no longer just done by physicians and nurses and physician's assistants. Okay, it's like everybody's involved now. Geneticists, molecular biologists, just everyone. Yeah, patients are now part of it. Like they have to be their own advocates and actually have to, do things to prevent development of illness. And then this last term really is based on the previous term. And it's basically saying not only do we need all these different types of scientists collaborating to improve the way that we understand and treat these genetic illnesses, but we need to set up systems in which the decisions aren't made by a single individual any longer. Okay? When you're starting to decide how you are going to treat or prevent a genetic illness, that we need to have systems in place where there are multiple people that are part of those decisions that get made. And I don't know, it's just such a wonderful sentiment. It's like, that just sounds great. Let's do it. Yeah, I mean, yeah.
How does that work?
That is a wonderful question. I don't know. And you actually get, there's a sentence in the textbook that says it's unclear whether or not this will ever happen. But it needs to.
It's just an idea.
It's an idea.
But it's
Yeah.
not.
I mean, there are some places that do it, and so basically what you're looking at is the idea here is to develop these systems in which healthcare is shifted from basically fighting fires to shifted in the hands of primary care physicians that are integrating information from all sorts of different professionals to try to develop a prevention plan. Yeah, try to develop a prevention plan. Yeah. Okay, cool. All right. we had this question before we ended on Monday, but we only got through one of the two slides dealing with this. How does smoking increase susceptibility to complex diseases? I think there are three more. So we have a specific gene, the ADRB2 gene, and at the 16th glycine, okay, in that gene, so the 16th glycine in the protein, we have a missense mutation that will switch that glycine out for an arginine. Okay, that's what this notation is saying, that at the 16th glycine residue, you'd have a missense mutation putting an arginine in there instead. And with that specific variant, you increase the likelihood of developing asthma by basically almost eightfold. Almost 8 times more likely to develop asthma. Smokers are almost 8 times more likely to develop asthma if they have this specific variant. Now notice this gene is for adrenergic beta-2 receptor, and these adrenergic receptors, these are part of the endocrine system, and they're the way in which cells bind hormones and then transduce and execute some response. And so that's just a little bit of context. Smokers are more likely to develop arthrosclerosis and type 2 diabetes. And the gene associated with this is called the CYP1A1 gene. And by this point now, I think we've maybe mentioned 7 or 8 very specific genes. And you might be asking yourself, well, do we need to know all these genes specifically? Which, that's a wonderful question, and I don't know the answer to that yet. But you could probably imagine what type of question would just be a wonderful question type to assess this, right? Like maybe have some specific genes on the left, or actually on the top, and then right below it, some specific, illnesses that they correlate with. You might see something, I don't know, very similar to that on the quiz that'll open up right when class ends. But your real question isn't like, is it going to appear on a quiz? Because you could just go and look in your notes and find the gene and where it is. But your big question is it going to appear on the final exam, right? Where you've got these, and I don't know the answer to that question yet. Smokers have a higher risk of Alzheimer's disease. And so this is just smokers in general, odds ratio 4.93, so an almost 5 times increased chances. Now, when smokers have a specific mutation in this APOE gene, that odds ratio increases to 6.5. So smokers become 6.5 times more likely to develop Alzheimer's if they have this specific gene. So that would be compared to people that are non-smokers without this gene. And so there you get some gene environmental interaction. And again, I told you this already, but this chapter isn't really big on giving you mechanisms. It's really big on just giving you specific examples of gene environmental interactions. But you can probably suspect what the mechanism will be if we're talking about a major environmental variable, that is smoking, and its influence on the expression of a gene. We're talking about probably something epigenetic and probably something semi-permanent, where we're talking about we're methylating the DNA directly, or we're methylating or acetylating histones. Yeah, Alyssa.
Does this mutation increase the likelihood of you getting Alzheimer's
Yes.
if you don't smoke?
Yes. Mm-hmm.
Okay.
Yep. We're going to talk about that in just a moment, because I think this gene is one of the best examples of antagonistic pleiotropy. So I'm glad you asked. And what is antagonistic pleiotropy again?
It's not impactful when you're younger, but it is when you're older.
Yes. Oh, that's so wonderful. Okay. So the next question, what are some important gene environmental interactions beyond smoking when the environment is something different? All right. So here's this gene again, APOE gene. So in this gene, there's a specific variant called the E2 allele. And the E2 allele reduces, so people with the E2 allele actually have a reduced what's the term I want to use? A reduced probability of developing Alzheimer's. Okay, people with the E2 allele at this APOE gene have a reduced probability of developing Alzheimer's. However, they have an increased probability of having really high serum cholesterol levels, which means they have an increased probability of developing atherosclerosis and other types of cardiovascular disease that correlate with high serum cholesterol levels. Now, the E4 allele of this is associated with an increased risk for Alzheimer's. And so what we're finding is that this E4 allele is decreasing in prevalence, probably because people with parents or grandparents that develop Alzheimer's are significantly less likely to have children, which is actually is true. I could show you some data on that. And so that this is basically disappearing because of, I mean, you could call it selection. It's hard to know exactly what do we call that if an individual is actually choosing not to procreate because of their genome, right? I don't know what you call that. Is that natural selection? I don't think so, but maybe it is. I don't know. Yeah.
Wait, so you're saying if you have a parent with Alzheimer's, you're less likely to have kids?
Yes.
Is that because they choose not to?
Yes. Yep. And so when you have some of these major genetic illnesses, you find that there's a very strong correlation between the number of offspring people produce and the appearance or the prevalence of certain genetic illnesses in their family. Anyways, that's another conversation for another time. But anyways, the E4 allele, the one that's associated with an increase in Alzheimer's, this is probably the best example of antagonistic pleiotropy. Early in life, it actually does a really good job keeping serum cholesterol levels low, which is probably a reason why it was selected for as human diets shifted from like hunter-gathering societies where you ate very little high-fat foods to different types of diets. And so now you have a benefit in this in that it lowers serum cholesterol levels, but late in life correlates with a significant increase in the development of Alzheimer's. Again, probably one of the best examples of antagonistic pleiotropy. Next part of the chapter was pretty interesting, and again, it wasn't really big on mechanisms, but it gave just a nice point to remember as we think about the interface between the genetic programming and the environment. And that's one of the biggest things that we can do is to shift our diet. And so it gave a couple of really good examples of how dietary changes can actually prevent the phenotypic expression of some of these complex genetic illnesses. Yeah.
So for the first one, that's if they smoke, right? There's a high.
No. That's and this is just in all individuals. Now there is a relationship. There's a gene-environmental relationship here with smoking, but this is for all individuals. Yeah, that's a good question. So we've already talked a little bit about this on Monday with PKU. So PKU is a monogenic genetic illness, and avoiding phenylalanine is a great way to prevent some of the phenotypes associated with that illness. But you still have the genetic illness, right? This is dietary changes to prevent ever developing the genetic illness. And so a couple of examples here where they gave specific genes. And basically, both of these genes, the ALOX5 and the MTHFR gene, both of these genes with their specific mutations correlate with the development of various cardiovascular diseases. And so this is a good example of where if you do a genetic health screen, and you find out that you have one of these specific mutations, these specific alleles, there are ways in which you can prevent ever developing that genetic illness by shifting your diet to something that's going to make it very unlikely for you to develop the genetic illness. And again, this chapter, it's not real big on mechanism, so I don't have like a specific mechanism to give you. I can find one, but it'll take pulling a paper, which I will do for our discussion on Friday. And then we'll get some more mechanism. This was a pretty interesting one too. It talks about that the genes for receptors that we find on white blood cells that are associated with targeting very specific materials on pathogens. And then this is, for example, TLR4, toll-like receptor 4. It's a specific receptor that we find on white blood cells. And this one targets a piece of a bacteria. And now, on top of that, it can also become confused and start to target other things, pollen, pet dander. And so what we find is that there are very specific mutations in some of these receptors. For example, TLR4. There's a very specific mutation in this that makes this receptor significantly more likely to bind something it should not bind, like pollen or pet dander. But you're like, okay, that's gene, although we're getting environmental because we're getting, non-pathogens. But what's cool about this is a lot of these mutations can basically be overcome by exposing yourself to the accidental antigen very early in life. And so it's like, okay, so there's a mutation in this gene that makes this receptor, instead of binding what it should bind, which is some piece of a bacterium, instead it starts to bind things it should not bind, like pollen or pet dander. But if you expose yourself to that antigen early in life, it will basically prevent your immune system from eliciting the response that it will do when it binds. It basically trains it to learn that what it's binding is not a big deal and to not trigger an immune response. And so you're like, this is so cool. And so the textbook talks about a specific mutation in here, and it's changing that receptor to react very heavily to certain types of pollen. But what you find is that people that grow up on a farm and are always exposed to that from the time that they're born, they never develop an allergy for it, even though genetically they should. They should, because the receptor is now binding it, but the cells have learned not to elicit an immune response to it. Alyssa?
Isn't this like the opposite of most allergies though? Because like most allergies, the more times you're exposed to it, the worse it gets.
I don't know if you can say most. There are many allergies that do work that way. That the more you're exposed to something, the more intense your reaction to it becomes. It has a lot to do with what is the mechanism by which you're activating the immune system. If the mechanism is antibodies binding to that, most likely your response to it is going to increase as you age, because you're going to make more and more antibodies the more you're exposed to it. But if the mechanism is something different, like one of these receptors, then what you may be able to do is to train it not to respond in the same way. Because these are going to typically be found on what we call our antigen presenting cells. Those that basically are what you might consider the first responders. They'll take it, they'll present antigen to T helper cells and will elicit, a what we would call a humoral response. And so it depends a lot on the mechanism. If the allergy is the result of like you've made antibodies against that antigen, then you're probably going to see it increasing in severity as you age. So I don't know if it's like a most or not. I haven't seen the numbers to see that, but it is common for allergies to get worse as you're exposed to them. Yeah.
And the ones that have to do with T receptor cells increase generally. Is there any way to stop those, or are they just like?
Oh, you can do whatever you want. Yeah, I mean, you can stop those, but if you're going to treat you're like, so we want to stop that signal later on. I don't know if we have a good way to do it specifically. Like if this cell triggers it, then don't do it. But if any other cell triggers it, do it. So yeah, I mean, you can shut down a complete portion of your immune system, but then you're at risk for not being able to fight a proper pathogen, right? And so that's what we do sometimes. Like if somebody gets an organ transplant or somebody gets a blood transfusion, we shut down certain portions of your immune system, and it makes you more susceptible for developing certain illnesses. Sid, did you have a question?
No, you answered it.
Okay. And then finally, not finally like we're done, but finally on this slide, we have the last gene I'll give you, ADH3. This is one of the genes associated with making our enzyme alcohol dehydrogenase, which is a complex enzyme that requires several different loci. But this specific locus, there's a specific mutation here that correlates with alcoholism and alcohol-related organ damage. And so this is, I don't know if you listen to the radio at all. They tell me when I'm listening to the radio, they advertise advertising on the radio, and they want to get you to, advertise your business. I don't have a business, so I don't have anything to advertise. But they say that radio reaches 85% of all consumers. I'm like, maybe that's true. I don't know. But anyways, they're advertising. There's a group at UCLA doing a study. They're doing a prospective study on this specific locus. And various treatments to try to prevent the development of an addiction to alcohol. And so I hear these radios all the time, but this is the locus that they're looking at, and then they're experimenting with different. So they're doing a prospective study on specific mutations there in the development of alcoholism, and then they're doing several controlled experiments to try to prevent the development of an addiction to alcohol. So I thought I'd share that with you because it's relevant to this. All right? Any questions about this question? We have one more thing to deal with, and it'll go fairly quickly. So without further ado, how do we do these gene-environment interaction studies? How do we actually do these? Because it sounds wonderful. We're like, well, cool. Like, just because you have this genotype, you aren't necessarily destined to develop the phenotype, right? That genetic illness. But the question is, how do we know this, and how do we do these? I would say probably most of them are done using retrospective studies. And then so what we're going to do there is we're going to recruit people with a specific genetic illness or a specific character. We're going to recruit people without that character, and we're going to give them all just this really extensive questionnaire, and not only just look at what is different in their genotypes, what is different in their genomes, but also what is different in their environment. Okay? What have they done differently, or what do they do differently on a regular basis to explain why they might have the genetic illness and somebody in our non-genetic illness group doesn't. These genome-wide association and interaction studies. Most of those are done doing retrospective studies. As you can probably imagine, we're not going to do a lot of these using prospective studies. Because the complicated thing about a prospective study is you're basically following people through their lives. And typically what you'll do is you'll follow people in one family and follow people in other families. And now if you're going to start to add other explanatory variables, other potential independent variables on top of their genotype, and you're studying this for decades, that's just an enormous amount of data. That's really complicated. So not a lot of these are done using retrospective studies. And then ideally, and a lot of this work now is done using controlled experiments. And a lot of these are even controlled experiments on humans, where we'll recruit people with the genetic illness, we'll recruit people without the genetic illness, and we'll change their diet, right? And so we're like, well, cool. Like, so if we're talking about, let's say, type 2 diabetes, and we have a suspicion that we can basically mitigate a lot of the pathology associated with type 2 diabetes and even reduce the severity of this by feeding somebody this type of diet, there's no real harm in shifting somebody's that does not have type 2 diabetes to a diet that you think is going to reduce the severity of type 2 diabetes, right? There's nothing really complicated about this, so we can do some really nice controlled experiments. Or we could do a controlled experiment where all you do, you recruit people with the genetic illness. Everybody's got the genetic illness. You randomly assign one group to your experimental group. You randomly assign the other group to your control group, and you change the diet of your experimental group. And you leave the diet the same of your control group. Okay? And you can do short-term studies. You're like, how do you control what people eat for a long? Was that what you were going to ask?
Yeah, well, like, what if the people in the controlled experiment want to get healthy all of a sudden?
Well, I mean, they can't, because they have to eat what you give them. They've enrolled themselves in a clinical trial. I mean, I guess you can choose to leave the trial at any point, right? You're not a prisoner there.
But why would anyone do that? Like, hey, I agree to keep on eating unhealthily.
Why would anybody do that? Agree to continue to eat unhealthily? I have no idea, Alyssa. Honestly, I have no idea why anyone would continue to eat an unhealthy diet. It's mind-boggling. I mean, it tastes so good. It tastes so good. You know, you get that dopamine release when you bite into that Doritos Locos taco. I mean, so it tastes good and it feels good. Yeah. Now, so for something like that, I mean, you're talking about probably, that one would be fairly complicated, but I mean, you can do it. Now, when you're doing, let's say you have a controlled experiment that's going to last 2 or 3 months, right? You're obviously not going to keep people inside wherever your clinic is for 2 or 3 months. And so you have to trust that they're sticking to whatever this diet regime that you've given them. But you know, it's like people have birthdays and it's like, what am I going to do? It's his birthday, right? I have to eat cake. I know I'm enrolled in this clinical trial and I'm not supposed to eat cake, but it's his birthday. And he wants to eat Taco Bell. I mean, what am I going to do? He even asked me to eat it, you know? All right. So I put this on here in this question of how do we actually do these studies. I put this nutrigenomics back on this because nutrigenomics is really starting to become a really big part of these gene-environmental interactions. Because it's one part of an environment that we can control very easily, right? You know, if you think smog or the exposure to certain pollen is triggering some genetic illness, I mean, that's fine, but some people are just incapable. I mean, they just can't go live somewhere else. It's not that simple, right? We're not talking about 150 years ago where, you know, you could just, the federal government would give you, is that Dr. Francis? Yeah. Where the federal government would give you a plot of land in the West, and they're like, please, we need people to go out there. And you're like, cool, well, we're kind of, you know, my son has asthma because he's breathing in all these fumes living in this dense city. So of course we'll go out there, right? It's not that simple. It's not that simple. But diet, you can change diet. You can change diet. So this is a very simple environmental variable to change. And so I want to talk a little bit more about nutrigenomics, some of the value of it. One is it allows us to suggest and develop personalized nutrition plans where we can basically not just have a general rule of like, you know, hey, have a little bit less fat in your diet, or, you know, have a little bit more of this specific amino acid in your diet. But no, we can study somebody's genome and we can make predictions about very specific things that they should eat to prevent the development of certain genetic illnesses. It's kind of cool. It also can help us, nutrigenomics can help us predict exactly what a shift in diet will do with regards to a specific genetic illness. So if we know that, you know, that this person's got one of those mutations that correlates with a increased risk for developing cardiovascular disease, And we could say, all right, well, we know, or we have a pretty good idea that if we shift this diet, we may be able to let them live long enough to die from something else, right? Which is really the goal, right? I mean, if you're a cardiologist, your goal is to help your parents live long enough to die from something else, right? And it's kind of depressing. I said that, and now I'm depressed. It just boiled down the entire healthcare industry, right? Allow somebody to live long enough to die from something else. All right. And then overall, the real focus of this, and why you're seeing a lot of money being invested in this, in nutrigenomics and in other gene-environmental interactions, is because this has a real potential to decrease healthcare costs. Which again is a cost that's not just burdened by the person who's sick, but it's a burden that is basically shouldered by everyone. Right? The amount you pay for your insurance premium oftentimes is not directly the result of how sick you are, right? It's directly the result of how sick everyone else is, right? So, anyways, yeah, I don't think about these things. You will soon. Many of you are actually going to go work in healthcare, and you'll think about these things way too often. And then the last point, and this is something we've already mentioned with two of our key terms, is healthcare now and the prevention of these genetic illnesses. It's really the work of healthcare professionals, dietitians, patients, geneticists, molecular biologists. I mean, it's a responsibility and the privilege of everyone to be involved in what's going on in the prevention of these complex genetic illnesses. All right, that's all I have for you. We finished it. And your quiz will open up in 4 minutes when class is officially supposed to end. And that's all. I'm leaving now.